In this video tutorial, I am going to discuss about beta oxidation of fatty acids in peroxisomes. Remember, the main bulk of beta oxidation occurs inside the mitochondria, and this mitochondrial beta oxidation is already covered in one of my previous videos. The key steps of mitochondrial beta oxidation are shown here on the left side. Peroxisomal beta oxidation is similar to mitochondrial beta oxidation in many ways, but there are critical differences that we must understand. In mitochondrial beta oxidation, fatty acids first get activated to acyl-CoA by acyl-CoA synthetase, which is located on outer mitochondrial membrane. Once activated to acyl-CoA, the molecule enters the mitochondrial matrix with the help of carnitine shuttle. This acyl-CoA then undergoes 4-step beta oxidation process. Now there is one major limitation in the mitochondrial system. This mitochondrial acyl-CoA synthetase works very well on short chain, medium chain and long chain fatty acids, but it does not work efficiently on very long chain fatty acids. And by very long chain, I mean fatty acids containing 20 or more carbons. I am not saying it doesn't work at all, but the efficiency is quite poor. As a result, very long chain fatty acids cannot enter inside the mitochondria. Now, this activation and transport process is hugely different in peroxisomes. Let me show you. Now, unlike mitochondria, which has two membranes, peroxisome has only single membrane. So, this is a peroxisomal membrane. Now, in this peroxisomal membrane, specific transporter known as ALD protein is present. Here, ALD protein stands for Adrenoleukodystrophy protein. This transporter allows easy entry of very long chain fatty acids. So here the first striking difference is that carnitine shuttle is not required. Instead, this ALD protein transporter facilitates the transport. The second difference involves activation inside the peroxisomes. Just like mitochondria, this acyl-CoA synthetase is also present in peroxisome. So acyl-CoA synthetase. But here it's of a different version, and this version is highly efficient on very long chain fatty acids. So these very long chain fatty acids are activated to their respective very long chain acyl-CoA. Now once the very long chain fatty acid is activated inside the peroxisome, the first step of beta oxidation, that is the oxidation step, is very much similar to mitochondria but with some key differences. In mitochondria, this reaction is catalyzed by acyl-CoA dehydrogenase, but in peroxisome, this same reaction is catalyzed by acyl-CoA oxidase. Although the enzyme is different, the overall reaction looks same. But wait, there is a subtle but very important difference. In mitochondria, this first step generates FADH2 and FADH2 passes through the electron transport chain to produce ATPs. But inside the peroxisomes, FADH2 does not pass through the electron transport chain. Instead, FADH2 transfers its electrons directly to molecular oxygen and gets converted to hydrogen peroxide. As ATP is not generated here, so the energy dissipates as heat. So as you can see, in peroxisomes, the first step does not generate ATP, whereas its counterpart in mitochondria does generate ATP. This is a major difference. Now, hydrogen peroxide is a strong oxidant that can damage various molecules. But luckily, there is another enzyme in peroxisomes called catalase, which breaks down hydrogen peroxide and converts it into water and oxygen. And just to balance this reaction, I need to write 1/2. Now, the second step of hydration and third step of another oxidation occurs exactly the same as mitochondrial beta oxidation. This third step, just like mitochondria, generates NADH plus H+, which is then transported to mitochondria to generate ATP. The fourth step is little bit different. This step is catalyzed by thiolase in peroxisome as well as in mitochondria, and reaction is also same. But the peroxisomal version of thiolase has one important limitation, that it cannot act on products that have 8 or less carbons. So what does this mean? Let's take an example. Suppose we have 20 carbon very long chain fatty acid. These undergo the first cycle of beta oxidation and becomes 18 carbon. Next cycle 16 carbon, then 14, then 12, then 10 and then 8 carbons. Once it reaches 8 carbons, this peroxisomal thiolase will not able to work, and obviously during each cycle acetyl-CoA gets released. So the final product of peroxisomal beta oxidations are acetyl-CoA molecules and 8 carbon acyl-CoA, that is octanoyl-CoA. What happens to this acetyl-CoA? It diffuses out of peroxisome and transported to mitochondria, where it undergoes Krebs cycle for complete oxidation and generates ATPs. And what happens to this 8 carbon acyl-CoA, that is octanoyl-CoA? The same story as of acetyl-CoA. It diffuses out of peroxisome and transported to mitochondria, where it undergoes further beta oxidation cycles. And by the way, as this 8 carbon acyl-CoA falls under medium chain category, it doesn't require carnitine shuttle to get transported across mitochondrial membrane. In this way, it seems that primary role of peroxisome is for shortening very long chain fatty acids so that mitochondria can efficiently work on this shortened fatty acid for complete oxidation. One more important thing, many authors says that peroxisomal beta oxidation does not produce ATP. However, this statement is not completely true, because I agree that the first step is different and does not generate ATPs. But the third step is the same in peroxisome. This third step generates NADH plus H+, and this NADH is transported to mitochondria and participate in electron transport chain and generates ATP. And acetyl-CoA generated with each successive beta oxidation in peroxisomes also diffuses to mitochondria to generate ATPs. The correct statement should be less ATP is generated by peroxisomal beta oxidation compared to mitochondrial beta oxidation. So this is how oxidation of very long chain fatty acids occur in peroxisome. Now there are two important disorders I want to discuss. The first important disorder is X-linked adrenoleukodystrophy. In this disorder, the ALD protein, which transports very long chain fatty acids inside peroxisome, is defective. This defect is genetic. The gene for ALD protein is located on the X chromosome, so this is called X-linked. And because it's X-linked, it is more common in male children. What happens if this transporter is defective? Obviously, very long chain fatty acids cannot enter peroxisomes, so there is a buildup of very long chain fatty acids in the blood. And these accumulated fatty acids destroys the myelin sheath of axon in many neurons. It also destroys white matter of brain and adrenal cortex, hence called adrenoleukodystrophy. Here, adreno is for adrenal dystrophy and leuko for white matter dystrophy. Now, this leads to loss of vision, behavioral disturbance, severe neurological problems, and adrenal insufficiency. The condition is so severe that death occurs within a few years. The second disorder is Zellweger syndrome, also called cerebrohepatorenal syndrome. In this disorder, there is a congenital absence of peroxisomes. Obviously, if peroxisomes are not there, very long chain fatty acid oxidation does not occur, and similar symptoms to adrenoleukodystrophy appears. In this disorder, mainly three organs are predominantly affected. These are the brain, liver, and kidney. So other name, that is cerebrohepatorenal syndrome. Cerebro refers to brain, hepato refers to liver, and renal refers to kidney involvement. Here, one thing must be clear, that peroxisomes don't only carry out beta oxidation of very long chain fatty acids. Peroxisomes are also important for alpha oxidation of branch chain fatty acids like phytanic acid and for synthesis of bile acids, plasmalogens, glycolipids, cholesterol, and dolicol. So, in Zellweger syndrome, as peroxisomes are completely absent, multiple other pathways are also affected, and therefore the severity in Zellweger syndrome is very high, and patients generally die at a very young age. Now, last but not least, some lipid lowering drugs, for example Clofibrate, stimulates peroxisomal enzymes. By doing this, beta oxidation of very long chain fatty acid is promoted, which helps decrease blood lipid level. So that's all about beta oxidation of fatty acids in peroxisomes. I hope that all the concepts are clear. If you still have any doubts or confusion, please write it down in the comment section below. Thank you.